By, you had it yet? Today?
Monday.
Oh.
It was so bad. I spent 5 hours on learning one set of slides and I couldn't remember. It was harder than.
Homework. Homework. Except for what I've never had.
Yeah. What kind of effect?
Dude, I'm really concerned. Why? Are we gonna put like 100% in the project? Like grades, it's like that's a block. I'm not gonna feel over yet. That's probably true.
Trust me on that. I'm working with it. Yeah.
So why would we say?
Hey, did you see the email I sent you about the?
I did,
Did you look interesting?
Yeah, yeah, I was planning on catching on now.
Okay, you can.
There's no possibility.
Hey everyone, how's it going? So we're going to talk today about more of the Gallant Lab Voxel Receptive Field models, which are how much did these are probably the most complicated models that I've spoken about in the class so far. So if there's anything that's not making sense or that seems confusing, I think it would be these ones. But now I know that people don't necessarily want to say like, hey, this is too confusing, I didn't understand this. But if there's any time when that would be a sensible thing to say, then it's now. And I'm actually going to get into some of the even more confusing stuff in this one. So do not be in the tiniest bit shy about asking about that. I guess that the main advantage of these models, although they're a little bit more confusing, they do have these very nice YouTube videos explaining them, which I think helps quite a lot. So I'm going to kind of go through some of that, go through some of the pictures that they show in that and try to explain and unpack them a little bit. And now I'm embarrassing myself because I need to run back up to my office and get that little Mac video dongle thing. Oh no, I don't. How is the literature searching, kind of finding what the key open question is for your project topic going? That's actually kind of one of those things that is much easier to say than to do. It can look very, very overwhelming at first when you say, okay, I'm interested in X, and then you start doing some search for X, and then you see there's literally 1,000 papers about it. Has anyone been encountering that? Yeah. Okay. So then the question is, okay, which of these 1,000 papers am I going to look at next? Has anyone? I mean, it's okay if you're still in the process of doing this, but has anyone feel like they've really kind of found a recent review paper that really kind of captures what they need? Because that's kind of the Rosetta Stone, if you can get that. Should I hit the other light as well? But if you're having difficulty finding useful, relevant kind of summary literature, then please don't hesitate to ping me because once you've found kind of one or two initial footholds, then that really helps a lot. Is anyone finding? I would be surprised if nobody is finding some difficulty. Is anyone? So don't be shy about this. Is anyone finding difficulty just kind of like you've looked for like the most? I'm interested in this broad topic, but I need to find something that's really going to kind of help me to get an overview of it, and I just cannot find something like that. Yeah, I'm pretty sure that at least 1 or 2 of you must be in that situation, because that's the most common situation when you're trying to do this kind of. I find myself in this situation all the time.
I had a holy grail moment.
Oh, you did? Okay, well, I want to hear about this. Okay.
Well, my interest, what I proposed is in the dorsal stream of vision. And
yeah.
I found a review that does exactly what I want to do in the ventral stream.
Ah, okay. Like this is the thing about self-motion, right?
Yeah. But
Okay.
this one is pretty much the same thing I want to do, except with object recognition.
Ah, okay.
It's within the predictive queue. So I want to do the predictive queue with visual motion, which is the dorsal stream.
Oh, okay.
And this one does an fMRI review of predictive cues with object recognition, which would be the ventral stream.
Oh, okay. Interesting. Is that who is that paper by actually?
Verga.
Okay. Oh, interesting. Okay, I don't know that paper. Oh, well, that's good. Well, if you're lucky, you might find something like that. But so that's perfect because you found something that's very relevant to what you want, but is not exactly what you want. So it leaves a kind of clear window of space for saying, okay, here's what we know, here's what we don't know, here's where it would be good to go next. Oh, thank you, Dave.
Okay.
That's perfect, having those middle ones on. Okay. So yeah, I mean, you may or may not find something that's quite that useful. Typically, you might find like 2 or 3 different things that collectively play that role. But it's very likely that you're going to experience this kind of initial sense of disorientation, just kind of hunting around, kind of groping around in a dark room. And do not be alarmed by that, because that actually means you're asking an interesting question. Okay, so anyway, so feel free to ping me about that, because and the other thing is that from chatting with you and emailing with you about the project ideas is actually one of you emailed me about this just earlier today, that my initial idea was everybody can either pick a publicly available data set to analyze, or write this kind of grant proposal-esque idea of here's an interesting study that would be worth doing, and here's how you do it, and here's why you should care. And what I'm starting to realize is that the branch of pick a publicly available data set and analyze it is that's great if you already have lots. I mean, this should have been obvious to me from the beginning. That's great if you already have a bunch of fMRI analysis experience, but if you don't, by all you're very welcome to try that, and it's my job to help you doing that. But that might be more than one class project's worth of material. That might be trying to bite off too much. And yeah, I think that should have been obvious to me beforehand. So, and I think that in emailing with you and interacting with you and chatting with you, I think that a number of you have also been coming to that conclusion. So, and I would say that that's completely fine, because in my opinion, the really interesting and difficult and important part is actually this question of trying to figure out, what is an interesting question, and how would we ask it, and why should someone care, and what do people already know about that, what do people not know, where is the kind of space to explore. That's really kind of what research is all about. The mechanics of actually doing the analysis is obviously very important, and is obviously good stuff to kind of get your hands dirty with. And we're going to be doing more of these computer practical sessions and getting our hands dirty with some of that. But to actually go through the full process of that analysis is very valuable endeavor, but is not necessarily. That's almost more like a kind of implementation detail of this bigger picture research question of how do we figure out what are the important and interesting questions to ask, and how might we address them. And I think that in fMRI that's actually a particularly important question, because less so these days, but for a long time in the field, fMRI was, and this is something we've talked about a bit in previous classes, fMRI was kind of getting a bad reputation. It was kind of a deserved bad reputation for just not really asking very interesting questions, for essentially just saying, here's some task X, and we stuck people in the scanner, got them to do task X, this is what lit up, and we're going to write a paper called The Neural Basis of Task X. And that's, you know, that's a step along the road, but it's certainly not the destination point along the road. And so I think that, you know, let's say that the field's gotten past that, or at least the most of the field's gotten past that. Then all of a sudden, your work is cut out for you a bit more, and actually trying to figure out, you know, well, if we're not just going to do that, what are we going to do? And luckily, there are plenty of things you can do. You can try and find relations to behavior. You can try and find some relations to underlying mechanisms. You can try and look at how representations are structured. So I think that being able to think about that, kind of getting some practice and kind of setting yourself this task of, you know, saying, okay, this is not a sufficiently interesting question, you know, but what would be? I think that's pretty valuable. So if you've also, and I know some of you have done this, if you've also gone through this period of thinking, this process of thinking, yeah, I'm going to, you know, take a data set and analyze it. Oh wait, that's actually, you know, kind of a big project. Maybe I should do something else. I think that's actually a very reasonable thought process. I know that some of you already analyze fMRI data, you know, from morning till night anyway, and they're in the process of doing this stuff anyway. So for you, it makes perfect sense to do that as a project, especially, and I think especially because you can tie it in closely with your existing work, and that's great. You know, that's wonderful. I think the more that a project ties in with stuff that you care about anyway, the better it is. I mean, there's no obligation. In fact, I would say quite the opposite, to feel that this project should be, you know, something just kind of totally sticking out on its own and not connecting with what you do in the rest of your time. I would say if you can tie it in, that's wonderful. For instance, I know that one or two of you are in the process of, say, applying for particular fellowships or applying for particular jobs, and I think that it would be great for this project to tie in with that. If I can, you know, if that can be a fit, then I would love to be able to help with that process. And I would think that, so I would say that there's a fairly expansive sense of what would count as an interesting project. What counts as an interesting project is something which, at the end of it, you can actually feel, yeah, you know, this has helped me to kind of get a picture of what are the open and interesting scientific questions in this imaging-related area, and how might one tackle them. And there's lots of ways in which that could go. Just as, you know, if you say, you know, what makes a good publication, what makes a good, you know, scientific research project, there is no single recipe. It's you know, just something which people go in, and at the end of it, they know something worthwhile that they didn't know at the beginning, right? There's one of the great wonders of science is that there's lots of different ways that can go. So anyway, so one way in which studies, fMRI studies, have really gone a lot beyond just seeing does some area light up, is this work of voxel receptive fields that we started to get into some of. You know, last time we got an overview of it on Tuesday, and we looked at a couple of, you know, these really nice videos that the Gallant Lab has made. And now we're going to go a little bit more into the kind of mechanics of what's actually going on here. So I think we got up to about here. So, but first of all, these are probably about as complicated models as we're going to cover in the class. So I'm pretty sure that these are definitely not. You know, I'm sure that I have not so far given a completely clear explanation of them. I'm sure that there's plenty of things that haven't quite, you know, been clear. So what is meant by the idea of a receptive field of a voxel? Does anyone want to give a little kind of mini description of that? Or how about what's meant by the receptive field of a neuron? I know that some of you do this for a living every day, so, but maybe someone who doesn't do this for a living every day could answer this question. It's like, this is not a trick question. I'm more, okay, well, okay, so this is good. I think we've reached a point that we've reached something that needs to be reviewed. So let me actually quit out of this and bring up the last lecture. Okay, so a receptive field of a neuron really has two, well, in this sense, in the Galois model actually has three characteristics, but the two key characteristics that are illustrated here are whereabouts in visual space. This is a visual receptive field, but there's equivalents in other sensory modalities as well. A receptive field is something out there in the world. Okay, so if you have a neuron, it's the aspect of stuff out there in the world that a particular neuron gets, that makes that particular neuron fire. So there's neurons in the head, especially in kind of higher level areas where it's not even possible really to say what their receptive field is. What's the receptive field of a neuron that's involved in rule-based processing in prefrontal cortex? I don't know. It's not even a well-defined question because there isn't just a kind of particular piece of space or type of stimulus out there in the world that will make that neuron fire. There might be lots and lots of things. It might be that the receptive field of that neuron is the business of dealing with the world, which is, kind of too broad to really count as a receptive field. So for a sensory neuron, the receptive field is basically the stuff out there in the world that makes that neuron fire. So for visual receptive field, there's two key characteristics of that. One is whereabouts is it in the world, and everything is in vision is with respect to where are you looking. So where is your fixation point? So if your fixation point, which is also the central part of the retina, is called the fovea, so if your fixation point is right here, then the receptive field of a neuron, say in this like purple quadrant 1 part of the brain here, is in this part of visual space. Okay, so that's the spatial aspect of a receptive field. Now this might jog some memories maybe. So how about a different aspect of a receptive field, a non-spatial aspect? Yeah.
It could be tuned to a particular feature, such as orientation.
Excellent, excellent. Okay, brilliant. So back when when we were looking at the Kamatani and Tong and the Hubel and Wiesel stuff, this was actually one of the key early discoveries in neuroscience. That there are these. Everyone figured neurons are going to be tuned to something, but no one could quite figure out what they were tuned to. And then they discovered, aha, it's not. Individual spots of stimulus don't really do anything. This is a diagram from this kind of short spot. You can shine. People were shining little spots of light. It wasn't really doing anything. The neuron's not firing very much. But then you move a bar, and suddenly it goes crazy. And in fact, actually, the neural recording apparatus is often hooked up to a microphone, so you can literally hear clicks every time the neuron spikes. You guys still do that? Do you have your neurons hooked up so you can actually hear them?
Oh, yeah.
Yeah,
Yes?
that's how you tell you're going from like gray to white to gray, like just
Yeah,
where,
okay.
where you are.
Okay, yeah. And it's very compelling. In fact, there's videos, I should try and find one of these on YouTube, but there's videos showing, with the audio track, a bunch of stuff being presented to a neuron, and all these different images flash up, and occasionally the neuron goes like click, 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 and then the right stimulus flashes up, and it goes like that, and it's quite striking. What a big contrast it can be. So that's two aspects of a receptive field: whereabouts in space it is, and what feature it's tuned to. In this case, orientation for visual neuron. There was actually one other type of feature that we looked at that's sort of related to what the orientation is, that we looked at as a visual feature of tuning that we looked at. Does anyone remember what that is? It's also built into this Galant type model. This was the slightly more obscure type of feature that you're probably more used to thinking of in terms of time rather than in terms of space. That's my little clue there. Does anyone remember what that one is? So if you have, so in space, in time, okay, so what, in time domain, what's the difference between, say, the note C and the note A if you play them on a piano?
Frequency.
Yeah, exactly. Okay, so what is the, so frequency means, in time it means literally means how quickly the air vibrations are going up and down, or in and out. Okay, in space, well, what's the equivalent of that in space? It's a little unintuitive to think of it in terms of space, in my opinion. But just because you don't encounter it, unless you're like an MRI physicist or an image processing person, you don't tend to encounter this that much.
Color?
Huh?
Color?
Oh, that is a very good point. That is a very good point, because that's actually the right answer. A right answer, but it's not the one I was thinking of. That's a very, very good point indeed, because, well, yeah, because color is, thank you, Molly. Okay, there you go. Color is actually based on different frequencies, which are different frequencies in space. What is the aspect of light that's changing between different colors? Yeah, exactly. Okay, so how spread out something is over time. Over space. Okay, so this idea, actually, this is a very, I like this as a metaphor. So it's more than a metaphor, actually. So this thing of spatial frequency is basically what I was trying to get to. But this is actually incredibly closely related to color in this interesting way that I actually had not thought of until you suggested that right now. So if you have a wave that's going up and down, many cycles per unit distance, this is kind of like, if you were to kind of plot what a blue light wave looks like over space. I hope I'm not saying something that's physically completely incorrect here. I mean, if you think of light as, like in this very literalistic way as a wave, let's not worry about that for now, okay? Then it would look kind of like this, okay? It's got shorter wavelengths, and the bars are closer, the ripples are closer together, basically. As you can, I don't know if you can see this, but these are not just bars, these are actually kind of, this is the grayscale version of a sine wave with a kind of Gaussian envelope. It's called a Gabor patch. Whereas if you have bigger wavelengths, then, which is a lower spatial frequency, right, the bigger wavelength, lower frequency, then it looks something like this. So this would be more like kind of, red light or infrared light or something like that. So that's actually a very good point. So in fact, color is actually a kind of spatial, color is a function of wavelength over space. Yeah. I don't know, does light at different colors like vibrate at different temporal frequencies too? It probably does, actually. That's falling off the edge of my physics knowledge, okay? But I'm sure some of you probably know that. Okay, so these are the key aspects of a receptive field. And for the receptive field of a voxel, it's the same idea that instead of just looking at what is the thing that makes a neuron fire, we say what is the thing that makes a voxel become more active. And this is also a kind of slightly unintuitive idea, because there's literally millions and millions and millions of voxels, of neurons in a voxel. So you might say, well, how could there be such a thing as the receptive field of a voxel? Well, it's really the kind of aggregate receptive field if you just average together the responses of everything that's happening in there. So when you measure the MRI signal from a voxel, there are a lot of neurons which are all firing, and they're all taking in oxygen, they're all getting extra oxygen sent to them. And all of that all together is going to collectively create this change in the MRI signal from that voxel, depending on how much oxygenated blood is in there. And so that kind of big amalgamated compound thing is what you're measuring when you say the receptive field of a voxel. And no one had really looked at the idea of a receptive field of a voxel until these guys, and they published a very influential Nature paper in 2008. So let's have a look a little bit at what is going on here. So so anyway, so I'm pleased actually in a sense that nobody gave the answer of what a receptive field was, because clearly that needed some revision. And so if you're not clear on what a receptive field is, then none of this is going to make any sense. So so is the idea of a receptive field a bit clearer now? Okay. How about, so that's what a visual receptive field would like. So it's useful to think the receptive field is stuff out there in the world. Okay? So what might say what might an auditory receptive field be like? Some of you probably studied this kind of thing. So the visual receptive field is, say, tuned to, a particular orientation at a particular spatial frequency, what might an auditory receptive field be tuned to?
Pitch.
Yeah, okay, yeah. And
Yeah, I was gonna say
yeah,
frequency,
okay,
yeah.
excellent. And what's another aspect? That's exactly right. What's another aspect of a sound that might influence how much a neuron is firing? I'll give you a clue.
Volume.
Yeah, there you go. Okay. So in fact, just as you can map out for a visual receptive field, how much contrast does there have to be? So this, I don't know if you can see this, but this little kind of mini grating patch here, Google patch, goes all the way from black to white, right? But you could have a lower contrast one that just kind of goes from slightly grayish to slightly whitish. Right? So similarly, so you can plot, it's called a contrast sensitivity function. You say for a given receptive field, how high does the contrast have to be on a particular grating or particular orientation to make this neuron fire? And same thing for auditory neuron. You'd say for a particular frequency, how loud does the sound have to be to make a neuron fire? I don't know if any of you have seen these graphs of how sensitivity to high frequency kind of falls off as you age. There's these ringtones that you can get that like teenagers can hear, but people like me can't hear. I think they're called mosquito tones, I think. So anyway, so this is kind of an example of one of these types of receptive fields, right? For the someone who's not a teenager actually would be able to hear that ringtone if you stuck it right by their ear and it was really, really loud. So it's this kind of compound function of pitch and amplitude. Okay, so it seems like receptive field is a key issue. What would a somatosensory receptive field look like? So one that you actually feel with your skin.
Temperature?
Huh?
Temperature?
Temperature would be a good one, right? So there's neurons that are responsive to that, especially pain-related ones. I'm not even sure how temperature is neurally represented. It's a very interesting question. But what would be the equivalent? So just as with the spatial map of visual receptive fields, there are different parts of the visual field with respect to where your eyes are currently fixated. What would a spatial map of receptive fields? What aspects of space would somatosensory receptive fields refer to?
Body space.
Exactly. Have any of you ever seen one of these pictures? It's called the somatosensory homunculus. It looks like this kind of weird, distorted person who's stretched out over your brain. Maybe I should. Well, I'll show that in a future lecture rather than trying to do a Google. I'm slightly nervous about on the video screen doing a Google image search for somatosensory homunculus. You never know what's going to turn up. But yeah. So the interesting distortions in that are the parts of your body that are extremely sensitive, such as your fingertips, look really, really big. Okay. So do you remember? I'm not going to switch PowerPoints now, but do you remember how the part of the around the fovea got represented by a really big chunk of brain? It's called cortical magnification. Exactly the same thing happens for the representation of your fingertips and other areas such as lips and tongue, also very large, in exactly the same way. So you have a big chunk of cortex devoted to representing your fingertips, much less than, say, representing your elbow or something, which is very unsensitive. Okay, so that's what receptive fields are. Okay, so this is going to, so what this approach does is it estimates receptive field for a given voxel. So what it does is, what they do is they say, okay, we've got, in this case, these three characteristics. We've got position, orientation, and spatial frequency, like how big and blurry is this patch. And so we're going to take a whole bunch of images. They just pulled a bunch of images off the web. For this first study, they just used black and white images because it helps to keep things simple. And they say, to what degree do each of these images actually excite activate these different receptive field channels. So, suppose I'm looking at a receptive field which is tuned to vertical things in this part of the image, right? Then if there happens to be a black and white edge, nice high contrast edge in this patch of the image, then bang, that's going to excite this receptive field. So if you take, remember we were talking about convolution, where you convolve you take a stimulus time course and you say, okay, each stimulus kicks off a hemodynamic response function. That's convolution in time. Okay. Well, this is convolution in space. You say, if I take this image and I kind of multiply each point of it by this kind of shape, this kind of on-off bar shape, what will I get? Well, if I take a uniform area like this and I multiply it by this kind of, well, I'll point to a bigger one because it's easier to see, this kind of on-off bar, well, the white part, which is like big numbers, is going to get multiplied by some uniform thing. The gray part, the black part, which is bigger negative numbers, is going to get multiplied by the neighboring area, which, because this is a uniform patch, is going to be exactly the same. They're going to completely cancel each other out. So, but if I take something like that that's tuned vertically and it happens to hit, say, this door edge here, okay, then the white part, and say it hits it just the right spot, then the white part will land on the white part of this, and you'll get big white positive numbers multiplied by big white positive numbers. And then the black part will hit the black part of this, and you'll get big negative black numbers multiplied by the big negative black parts of the filter. And then that will be even more positive stuff. So you'll get lots and lots of response to the edge. So this little patch here is basically a little vertical edge detector. And we have things exactly like this in our primary visual cortex. So that's why this is the model that they pick. Okay? So they're basically saying, okay, if I have a whole bunch of oriented edge detectors in my visual cortex, which are which are tuned for particular parts of space, particular orientations, and also how wide the edge is. Basically, it's not quite an edge detector. I mean, it's a spatial frequency detector, but you can think of it as an edge detector. It's a reasonable approximation. So, this is the big ones are tuned for kind of big, blurry, large scale changes, like say a big wide door next to a big wide patch of wall, and these thinner ones are tuned for much more rapidly changing things, like say a lamppost against a black lamppost against a white sky or something like that, something thin. That would excite one of these. Okay? So you say, okay, how much does, I'm going to take this image and convolve it with all these patches, which will tell me how much does this particular image, make each of these patches light up, in effect. Okay? And your primary visual cortex it's doing a bunch of other stuff too, but in large part, that's what your primary visual cortex is doing. Okay? That's what, 40, 50 years of visual neuroscience has shown. It turns out it's doing all kinds of other things as well, but for a first approximation, it's taking images that come in and land on your retina, and the neurons that have different receptive fields are saying, okay, I fire when I see vertical edges in this part of the visual field that are of this kind of width. Oh, there's one in this image, bang, I'm going to fire. Okay? So you do that for this whole kind of bank of filters, and then you get the kind of sum number out that says this is how much everything's firing. If those of you who are really into image processing will also notice that, in fact, these are like two phase-shifted versions of the same filter, and it's integrating across different phases but you don't have to worry about that. Okay? But basically, you're looking for how much of these images exciting edge detectors of all different shapes and orientations and sizes. And then you say, okay, that's basically our model of how visual cortical neurons would be responding to this image. If our voxel that we're looking at has a response that seems to be made of neurons, with these particular types of tunings, then suppose we find a voxel which starts to light up quite a lot when we show this particular image. Okay, then we'd say, okay, well, there could be a whole bunch of different things driving this voxel. It might be that this voxel, really likes black vertical lines right here, but you're showing the whole image, right? So maybe this voxel is actually being excited by this part of the image or that part of the image. Okay? But we know, that at least in early visual cortex, that voxels in particular locations tend to be looking at only fairly small patches of the image, just because there's this spatial map. Remember the thing that I showed you before? So there will be lots and lots of different orientations present in that voxel, but it will tend to be located around a particular point in space, assuming that your subject is doing a good job of keeping their eyes still. Okay? So you can actually, so if you show lots and lots of images and record the activation from voxels and take each of these images and kind of apply these filter banks to them and say, okay, to what degree is the image stimulating these filter banks, then you can say, okay, that's, given that I know how much my voxel responded to each of these images, I can kind of fit this model and say, okay, I think this voxel on the whole responds like a filter bank that's like this. Right? You know, this voxel looks like it's really interested in, you know, this range of orientations in this particular location, this particular spatial frequency. In fact, let me show you a picture there. So that's what they're doing here. So they're taking the image, showing it to the brain, and looking at the activation across a whole bunch of voxels. Okay, so what does one of these receptive fields end up looking like?
I have a question before you move on.
Yes, yes.
I think it was on Tuesday I got the impression that most of these types of studies don't control for eye movements.
I may have made a mistake if I gave that impression, because eye movement, well, it depends what you mean to control for eye movements. Now, if you really want to control for eye movements, then you actually have to have an eye tracker in the scanner. And I'd have to check the methods section to see whether they actually did that or not. But they definitely required their subjects to fixate properly.
How do they know that their subjects are fixated?
You know, so that's right. So you know, this is a good question, so we should look at the method section. So first of all, they basically just did this on themselves. So you remember in the video, they said, you know, we took 5 people and we scanned them for several hours each, right? Well, no normal subject would ever put up with that. Often, these kind of studies are often done on the experimenters. Now, there's some types of studies that you just cannot do on yourself, okay? If you're going to do a memory study where, you know, you remember we looked at one of these where you divide the stimuli afterwards into the ones that people remembered and people forgot. You can't do that on yourself because you've seen all the stimuli, right? You can't possibly see one of these things for the first time. Similarly, anything that involves some kind of problem solving, or, you know, the subject going from one cognitive state to another cognitive state, you can't really do that on yourself, okay? But if it's something like, you know, look at this image and, you know, see what it does to your brain, you can do that to yourself. It doesn't, unless you've done something very strange, it shouldn't really matter that much that you've seen this particular image before. I mean, it depends upon exactly what kind of task you're doing. Similarly, you know, if you're just getting someone to listen to, say, a bunch of speech sounds, you know, you can do that on yourself, at least for testing purposes, right? You might, for piloting, you might not necessarily want to use that data, but for these kind of low-level sensory tasks, it's okay to do it on yourself. So I'd have to check in the methods. So they probably, you know, if they really wanted to check that they weren't cheating, if they were just bringing people off the street, then they would have definitely needed an eye tracker to make sure these people really were keeping their eyes steady. Because you can't just trust, like, it's kind of hard to do that. I mean, you probably know this from your own experience. But if you're doing it on themselves, they probably actually kind of knew that they were fixating properly. But that's very, very important, because there's no way. So this is a map of the spatial, so this is the visual field, okay? This is a map of the spatial, the part of space that this particular voxel tended to respond to, with fixation being here, okay? And so it's kind of like a little bit to the left of fixation, but kind of like on the, you know, level with fixation in this horizontal direction. And so this map would be completely impossible to create if someone's eyes were not pretty much fixed here in a nice, reliable way. I mean, nobody's perfect. Everybody blinks a bit or, you know, looks away sometimes. But if you scan someone for 5 hours, it doesn't happen, right? But yeah, it's just, you know, maintaining fixation. Well, those of you who work with monkeys know that the first thing you have to train a monkey to do is to depending on what kind of test it is, to maintain fixation. And I think it kind of takes a while to train a monkey to do that, right? But you can really monitor that. I mean, you know, these monkeys' eyes are very closely monitored. But anyway, even for people, it's not that easy either. So this is what a receptive field ends up looking like. So the space part is the easiest part to understand, right? And this is really just a function of whereabouts in the brain, whereabouts in this visual cortical map is this voxel. You could actually, to a reasonable degree, tell where in space the receptive field would be, even before you'd actually, I mean, to some degree, you could actually tell, you could at least tell which quadrant it would be in before you've even run the study. You can say, if it's in this part of the brain, you know, and this person is keeping their eyes in the right place, it's got to be in this quadrant. To say exactly where in the quadrant, then you have to kind of measure it. But, okay, so here is the slightly less clear bit. So what orientation is this voxel tuned to? Now remember in the Kamatani, when we're looking at Kamatani Tong, we're saying these orientation maps are much, much, much finer grained than the size of a voxel. I mean, an order of magnitude, a couple of orders of magnitude finer grained. But because they're kind of swirly and irregular, it can be the case that there tends to be a little bit more, just by chance, a little bit more of a concentration of particular orientation, particular parts of the orientation map in this voxel than other parts. So that means that, you remember how we had these kind of histograms of orientation responses, and some of them were kind of flat, but some of them were kind of a bit pointy, meaning that there was some preference for particular orientation. So this is basically just like that, except it's, you know, it's drawn as a line graph instead of a histogram. So this voxel happens to have a little bit of a preference to orientations of 45 degrees. Okay? But notice that it responds to some degree to all of the orientations. It just responds a little bit more to 45 degrees, which is what you'd expect because there's neurons tuned to all different orientations in there. Whereas, so this is you know, the baseline. Everything is way above baseline here. Whereas that's quite different from the spatial map. For most of the areas of space, because the person's really keeping their eyes in the right place, you basically get no response at all, except a little bit of noise, and then you get lots of response. So this is a very peaked distribution here. Okay, so a neuron's a voxel. Yeah, a neuron may be very tightly tuned to particular orientation, but a voxel, because it has lots of different neurons in it, will never be very tightly tuned for a particular orientation because the voxel's just too big. I think there was a question somewhere, but I may have just, no, okay. And then you can do a similar thing for spatial frequency. This is that slightly less intuitive measure. So it turns out that this voxel seems to like, I don't know if you can see this, but it seems to respond more strongly to higher spatial frequencies than to lower spatial frequencies. Another way of saying that is it likes kind of fine-grained sharp edges more than it likes big blurry edges. Which is actually a very common feature of early visual cortex. They tend to be quite small receptive fields. To be tuned to a really big blurry edge, you kind of need to be a bigger receptive field, really. And this little 2D plot is actually just these two plots kind of multiplied by each other. So in this direction, you're plotting this, and in this direction, it's plotting this. So you can see that you've got a kind of peak around 45 degrees, and you've got a peak in this direction around higher spatial frequencies. And the fact that it's kind of quite uniform in this direction means that the fact that this neuron likes higher spatial frequencies is true across the orientation. So whether you're looking for, you know, edges that are like this, or this, it kind of likes fine-grained edges. Okay? Which again is exactly what you would expect for a primary visual cortex neuron. So these are the three aspects of Voxel's receptive field. So does that seem like it's sort of making sense? Because that's what these models are built out of. So this is like the key building block. If there's any part of this that seems confusing, I would say it'd probably be the spatial frequency bit, but that's just me. But, okay, so you've gotten these receptive fields, and then here's a nice bit. Okay, you can say for a particular this is how having a model of responses to visual images, not just these particular visual images, but a model of responses to visual input in general. This allows you to extend beyond the stuff that you've already seen. Okay, this allows, remember we showed this picture, I think I'm actually going to bring this picture up, hang on a second. because this is an important picture. This one here. Okay. So the key advance of this K et al. Paper from Jack Gallen's lab, and it came out more or less the same time as paper by Tom Mitchell that we're going to look at in a week or two, looking at